Thank you so much. It really is a privilege to be here and share the stage with leaders of the field like Dr. Becker and my good friend Dr. Wilton Williams. And in fact, I was talking with Wilton yesterday that maybe we could jazz this session up a little bit if we came in some Barbie Oppenheimer themed uniforms. And here I am in my Oppenheimer black and gray, and apparently Wilton had second thoughts and has just a little touch of pink on his tie. Regardless. It really is a privilege to be here, and I would like to begin by acknowledging the Yurgera and Turrbal peoples, the traditional owners of this land, and to honor their elders past, present, and emerging. And I'd like to continue by talking a bit in a more general overview about strategies for using antibodies for HIV cure. And so as an outline for this talk, I want to start by addressing sort of the specific challenges and opportunities of using antibodies for HIV, then think about practically what are the clinical outcomes or the goalposts that we need to achieve in order to successfully use these antibodies, and then end with current strategies that are currently being tested that have some promise to translate antibodies to actual cure eventualities. So, for context, we in the infectious disease community recognize that antibodies are central to antiviral immunity across a range of viral pathogens. When we think about vaccines, antibodies are the correlate or the co-correlate of protection in the vast majority of FDA-approved licensed vaccines. In addition, when we think about virus cure or clearance, there's an increasing recognition that neutralizing antibodies drive virus clearance. Even in hepatitis C, a very diverse RNA virus that in theory should be hard for antibodies to address, we now know that neutralizing antibodies are often the determinant of spontaneous cure or spontaneous clearance, both in humans and in animal models. However, in HIV, this is not the case at all. And that is because, perhaps more than any other viral pathogen, HIV-1 is highly evolved to evade neutralization. So if we think about neutralization is binding of the functional HIV-1 OMV trimer so that it cannot go on to infect the next cell, we realize that HIV has many, many strategies to block this. First of all, in this schema of a virion, we can see that HIV, oh, the mouse works. We can see that HIV displays many decoy immunogens. So we have little GP41 stumps, we have GP120 monomers or dimers, but these distract from the functional OMV trimer that the virus is trying to protect from the antibody response. And then when we look at that trimer, as Wilton was describing to us, it's covered in a glycan shield that protects it from the immune system. And its conserved epitopes are deep within recessed pockets that are very challenging for antibodies to access. Secondarily, we know HIV specifically targets CD4 positive T cells, both directly and indirectly. And we know that these T cells are necessary to encourage or to have antibody maturation process. And with limited CD4 T cell help, that delays or stalls the maturation process. And finally, we know that HIV-1 is an incredibly diverse virus. This virus rapidly mutates, it tolerates diversity, so with any one individual living with HIV, this virus rapidly diversifies. And we think about the viruses circulating within a community or within the world, we recognize that extensive diversity is very challenging for any one antibody to be able to conquer. So what we have is that the autologous neutralizing antibody response in HIV-1 infection is delayed at every single step. The first antibody responses are actually cross-reactive from gut microbes, leading to binding antibodies to GP41, one of these decoy immunogens. We see our first de novo or new responses arise about a month after infection, where we see targeting with binding antibodies to GP120, again, not the functional trimer. These binding antibodies allow us to diagnose HIV, but they do little to clear the virus or to functionally move towards cure. And it's not until about 3 months after virus transmission that we see the development of neutralizing antibodies. Now, these neutralizing antibodies that arise at 3 months can be quite potent, but by the time they show up, the initial infecting virus, the transmitted founder virus, has long since escaped from that antibody response. And we see that throughout the natural history of viremia. The antibody response is slow and the virus rapidly escapes. Only after that cycle goes on and on for many, many years, in a subset of individuals, do we see the elicitation of broad or cross-reactive neutralizing antibodies, these BNAbs that are so desirable as reagents and as models for vaccination. But even in that scenario, the virus that is circulating at that time has long since escaped from being sensitive to that evolving BNAb. So the theory of using antibodies, BNAbs or otherwise, in HIV is to change that relationship between that slow antibody response and that fast virus escape. And the goal is to use our best antibodies at a clinical juncture before that virus is able to escape. So prevention, we're talking about delivering BNAbs or immunized vaccine strategies before any individual is confronted with a vaccine or with a virus challenge. For cure, we think about using antiretrovirals to block that virus from further escaping, and then delivering our best BNAbs or using immunization to drive that antibody response to be bigger and better than the virus that was previously trying to escape from it. Okay, so when we think about how we might use antibodies in people living with HIV, I think it's valuable to think about interventions in people living with HIV on a sort of treatment-cure spectrum. So on the left here, we have antiviral therapy, where we mean to suppress virus and return people to health. But as we move this continuum to the left, we're looking to chip away at the reservoir to boost host immunity so that we could potentially have sustained virus suppression off medicines or a fully eradicative cure. Our current antiretrovirals are amazing. They are highly effective, they're extremely well tolerated, but they are antivirals and they do not move us towards cure. Antibodies, in addition to having lots of desirable properties like long activity and low toxicity, also have specific properties that allow us to consider them in terms of cure. So in particular, I'm talking about effector functions whereby antibodies can help clear virus-infected cells. And because these are immune agents, we can enhance immune responses with these antibodies. So to dig into those properties just a bit, if we think about reservoir reduction via effector function, what we mean is we have a virus-infected cell that expresses any virus properties on its cell surface. That antibody with its Fab region can bind to that virus antigen and then recruit immune cells, NK cells, macrophages, to help clear that infected cell away. In addition, we have a concept called the vaccinal effect. Now, the vaccinal effect is very well established in cancer biology. It is much less so proven in HIV cure. However, we think that the concept that virus and antibody can bind together, form immune complexes that interact with antigen-presenting cells or dendritic cells, and thereby boost HIV-specific immunity is a really exciting potential. And that's one of the goals of the field, is to try to really test if and how this can happen in HIV cure. Okay, so if we have these wonderful properties about antibodies, what are the realistic clinical endpoints or goalposts that we need to achieve to be able to use these antibodies in real life? In my mind, we have to demonstrate antiviral activity in people in vivo. We have to show virus suppression akin to what we see with antiretroviral therapy, and then we have to move towards drug-free virus remission by using this enhanced immunity and the clearance of reservoir cells. So this first goalpost, or antiviral activity, was accomplished about 10 years ago. So multiple studies were conducted, but here I'm showing a representative figure from a study led by Dr. Marina Kasky, in which a single infusion of a CD4 binding site BNAb, 3BNC117, was given to viremic participants, and the viral loads were followed. And what you can see here is a more than 1 log drop of viremia in these individuals that lasted for a couple weeks. Now, this is a genuine antiviral effect. It's similar to what we see with single small molecule antiretrovirals, but it is transient, and we did see virus resistance emerge. But after sort of demonstrating this first goalpost, the field moved to see, can we demonstrate virus suppression for durable periods. And we did this largely through treatment interruption studies, or ATI studies. And these are essentially switch studies. You take an individual who is suppressed on antiretrovirals and say, can we switch the small molecules to antibodies and maintain virus suppression? So what I'm showing here are, again, Kaplan-Meier curves showing time to rebound in individuals who switched from antiretrovirals to, on the left, VRC01 in 2 different studies, and on the right, the more potent 3BNC117. And what you can see is there's a slight shift to the right of these Kaplan-Meier curves, indicating a modest increase in the time to rebound. And the shift depended on the potency and the breadth of the antibody. But again, we recognize that many people broke through early, and pre-existent resistance was common and an important challenge. So to dig into this, in the ACTG study previously discussed with VRC01, we asked, in these individuals who had breakthrough virus very early with high concentrations of antibodies, what was going on with the viruses to explain this? So we sequenced and cloned virus from a preserved sample collected years ago before these individuals started antiretroviral therapy, and then at the time of rebound. And if we sequence and clone those viruses and test their sensitivity to VRC01, what we can see when we compare in black the pre-art virus and the rebound viruses in red and yellow is that, yes, rebound virus was resistant to these antibodies, but in fact, that baseline resistance existed years before, prior to exposure to this antibody. So baseline resistance remains a barrier, even with some of our better antibodies. And so with that, the development of screening assays became a goal in the field. Now, I work in this particular area, but I think it's important to recognize, yes, it would be very helpful to have a predictive assay, but it is very hard to sample the diverse reservoir within any one individual and to have confidence that you have not missed any of the small minor variants that may contribute to success or failure with this BNAb agent. However, at this point, the field has one assay, the PhenoSense assay by Monogram Biosciences, that is CLIA and CAP certified, and it has currently been used in multiple clinical trials. And so we at the ACTG partnered with Monogram to validate and to test this assay's performance. We looked at how this assay performed in different sample types, on cells and in plasma. And then we looked at different durations of antiretroviral therapy, 1 year, 3 years. And we showed that these results were incredibly tightly correlated. So this assay was very nicely controlled over time and over sample type. However, when we used this assay and we retrospectively looked at ATI BNAb studies, the pre-screened values were not able to convincingly predict whether that antibody would successfully suppress that virus for a period of time. And so that is a bit disappointing. In addition, this assay, although relatively high throughput, does add logistical challenges in terms of time and cost and the number of individuals that must be screened for a study. And so that is also a challenge. Now, this assay continues to be used in multiple studies, as well as not used in comparative studies, and new assays are in development that are sequence-based or with different assay platforms, and these all will be judged in the next few years as we sort of percolate and think about how these assays can be applied. But my hope is that the pipeline with new and better BNAbs and BNAb combinations will supersede the need for these screening assays. And in fact, we have shown remarkable improvements with combination BNAbs and their ability to elicit durable virus suppression. So I'm going to show here 3 Kaplan-Meier curves from studies looking at combination BNAbs at treatment interruption, these switch studies that have been either presented or published in the past year or so. And in general, what we're looking at is 3BNC and 101074, a CD4 binding site and a V3 glycan BNAb together, multiple doses given at ATI, and we can see that there's remarkable levels of suppression. Between 71 and 83% of these individual participants maintain virus suppression for 6 months of continued antibody dosing. I'll mention that on the left and the right, there was no pre-screening done in these studies. And in the middle, the BEAT2 study that I was involved with, we did do pre-screening. There's slight differences, but I think all of the nuances of the pre-screening will continue to be worked out. But regardless, I think that we have seen durable suppression within sensitive hosts. And the challenge for the field is how can we improve our BNABs and our combinations to expand the number of individuals who are, quote, sensitive hosts, so that we can use these antibodies much more broadly. But I think it's also important to recognize in the studies to date that have been using BNABs on antiretroviral therapy or at ATI, we really see minimal evidence of either substantial reservoir reduction or substantial immunomodulation. And those are the qualities that we're most excited for antibodies for cure. So what is the field doing in terms of new strategies to enhance those cure potentials? Well, there's 3 strategies I'm going to mention that I think hold promise. The first is combination immunotherapy. And specifically, we're talking about combination immunotherapy in the pursuit of this shock and kill strategy, whereby we try to use latency reversal agents to have latent cells show themselves to the immune system by starting to transcribe and translate virus. And then we use BNABs or other agents to help clear the reservoir, those infected cells that have shown themselves. And we also use BNABs and enhanced immunity to suppress that virus that might be coming out of those reactivating cells. Now, there are many, many immunotherapeutic agents being used in these combination studies. There's a lot of different trials in development, but I'd like to highlight one recently presented study that has used a specific group of these combination therapies. So Dr. Michael Peluso presented these data at CROI and also on Saturday at the IAS Cure and Immunotherapy Forum of the UCSF AMFAR study looking at combination immunotherapy. And in this study, they used 3 modalities: a DNA MVA vaccine strategy meant to boost CD8 T cell responses. In addition, they used BNABs plus a TLR9 agonist meant to enhance the ability of that antibody to clear the reservoir, and then another dose of 2 BNABs right before treatment interruption to suppress virus and allow the immune system a little time to continue its good work. So these data are preliminary, but as Dr. Peluso has presented, the rebound kinetics of these participants is interesting. They did see expected exponential rebound in some participants here, but in others, what they saw was a much more blunted or delayed or less robust low-level viremia. And this suggests that this combination of enhanced T cell immunity and enhanced BNAB virus reservoir reduction may have enhanced the ability of these individuals to control their virus after these interventions are over and off drug. Okay, what's the next strategy to talk about? Well, this is the concept of using broadly neutralizing antibodies and other interventions at the timing of ART initiation. So there's 2 compelling reasons why I think this is exciting. First of all is the concept that reservoir formation largely happens around the time of ART initiation. So in a beautiful study done by the Caprisa cohort led by Abrahams and colleagues, what this group did was they followed a cohort of women who were diagnosed in early disease, followed for viremia for many years, and then suppressed on antiretrovirals and had their virus reservoir sampled years after suppressive ART. And what they found was the reservoir sequences, shown in pink here, aligned most closely with the viruses that were replicating at the time of ART initiation. This suggests that the stabilizing properties of antiretrovirals may have led to that durable virus reservoir forming. And it suggests that this time point may be a really good one to disrupt reservoir formation. So the second reason is if we think about that timing of ART initiation, there's actually a lot of virus in the system. Just before and after ART is started, we have high levels of virus that could bind with antibody, make these immune complexes, and maybe have a very effective vaccinal effect because of that. And so the concept of using these together at the time of ART initiation is to block the reservoir and then to enhance that vaccinal effect with immune complex formation. So the eClear study, led by Dr. Ole Sogard and his colleagues in Denmark, tested just this hypothesis. They asked, can BNAbs plus or minus a latency reversal agent at the time of ART initiation, can this limit the formation of the reservoir and enhance immunity? They studied 4 arms, ART alone, ART plus a BNAb, ART plus an LRA, or LRA plus BNAb. They followed for a long period of time, and then participants had the option to undergo a treatment interruption. So remarkably, what this study showed was a positive signal for each of the pre-specified aims. So they saw evidence of increased clearance of HIV-infected cells. What this here is virus decline, and this second phase of viral decay was faster in those who got BNAbs. In addition, HIV-positive cells declined faster. Secondarily, they looked at HIV-specific CD8 T cell responses, and what they saw in BNAb recipients with sensitive virus was a greater frequency and greater activity of these cells in those individuals. And in the limited number of individuals who underwent a treatment interruption, what they showed in individuals with sensitive virus who got BNAbs in these light blue lines is a much more muted viral rebound, where there's evidence of virus control post-treatment. This is long after the antibodies have left the system, and it suggests that the immune responses may have been substantially altered by this intervention. So this study, I think, is the first step and provides cautious optimism for the field to try to validate this with other trials. And right now there are multiple studies either ongoing, such as the Rio study exploring these concepts, as well as other studies in development, like several from the ACTG looking to test BNAbs at ART initiation, both in the Western world as well as in sub-Saharan Africa. So the final strategy that I want to mention is therapeutic antibody-based vaccination. And this leads back to all of the exciting work that Dr. Williams just told us. The goal here is to look with people living with HIV, suppressed on antiretrovirals, and ask, can we expand their autologous antibody responses so that they could suppress their own virus? And this is really possible because of the major innovations that have happened in the vaccine field. In the HIV vaccine world, we have amazing new stabilized trimers, adjuvants, dosing strategies, mRNA and lipid nanoparticle technology that allows us to dream big when it comes to what our vaccinations can do. There's also new evidence in my mind that's exciting about maybe the antibody response on antiretroviral therapy is a little bit more active than we had previously thought. So for example, Dr. Jonathan Lee from Harvard recently published a paper in which he characterized individuals who started antiretrovirals, and he showed that their autologous antibody response rose substantially on continuous ART treatment and rose even further after treatment interruption. In addition, a recent case report about individuals with post-treatment control from Dr. Taeuk Chung from the NIH showed that in this one example of an individual who stopped their antiretrovirals and maintained suppression for many years, this suppression is tightly correlated with their autologous antibody response. And similarly, work from my group in collaboration with Dr. Sugi Lee from UCSF looked at a cohort of early ART initiators, and what we showed was that the autologous neutralizing antibody response increased in potency over months or years of suppressive antiretroviral therapy. And similarly, in participants who started antiretrovirals with a diverse virus population, the antibody response not only increased in potency, but it increased in breadth. And so initially, we only targeted some of the variants, but over time on antiretrovirals, the antibody response began to target more and more of these diverse viruses. So this suggests, at least in early ART initiation and potentially elsewhere, the antibody response may be primed and it may be receptive to using these novel vaccine approaches in order to boost and expand the potency and breadth of that individual's antibodies. And so this is an early stage. There are several strategies in trial right now, and they really, if you recognize this figure, this is from Dr. Williams, but this same strategy that's being used in prevention is now being adapted in people living with HIV. And the question is, these priming immunogens, etc., that have shown such success in eliciting rare BNAb responses in naive hosts, what will they do in individuals who already have an HIV-specific antibody response? And so our first questions are, what B-cell lineages are expanded? Do we see changes in autologous antibodies or in broadly neutralizing responses? But ultimately, we need to take these approaches and move them further and ask, can we expand that autologous response to help control each individual person's virus population? And I have optimism that we can do that over time. Okay, so in summary, I hope that I have been able to give a broad overview of the potential for antibodies for HIV cure. And though antibodies are largely ineffective in natural history of HIV, I think if we use our best antibodies prior to virus escape, there is real potential. We need to exploit antibodies' unique properties, including their immune activity and effector function, but we also need to continue the pipeline to develop antibodies with better potency, breadth, and important functionalities. I think we have demonstrated certainly that these antibodies have antiviral activity and that we can maintain virus suppression in sensitive hosts. We need to expand what sensitive hosts means by making these antibodies broader and more potent. And then we need to focus on our current objective, which is using these antibodies to induce drug-free remission via our strategies that are in trials right now, as well as new, thoughtful and creative strategies to use these powers of antibodies in order to engender cure. So with that, I will say thank you very much for your time, and I appreciate this opportunity.
I'm sure you will agree with me that there is much to be excited about. I hope you're as excited about BNABs as I think we all should be after two magnificent talks. So well done to both of our plenary speakers this morning. I would ask you just to hold on. There is going to be an award ceremony now, so please do keep your seats. But I want to also just give a shout out that the International AIDS Society believes that a vaccine and a cure are possible, and they have two initiatives. The Global Vaccine Enterprise is ongoing, and the Towards a Cure initiative is also ongoing. So I ask you to continue to, I think Katie had the call to action, dream big. And believe that this can happen. So thanks so much for everyone's attention today, and please hold your seats for the awards. Thank you.
Please welcome to the stage Annette Sohn and Kenneth Meyer to present the JIAS Impact Awards.
Good morning, colleagues in the room. Good afternoon, good evening to colleagues online. We're the editors-in-chief of the Journal of the International AIDS Society. I'm Ken Mayer.
And I'm Annette Sohn, and we're pleased to announce the recipients of this year's JIAS Impact Awards, acknowledging their high-impact publications in our journal. 3 articles were selected by our editorial board and deputy editors, and each first author is awarded an in-person registration at a future IAS-organized conference. The award is sponsored by the International AIDS Society.
Our first award goes to Kelika Kanda and Tiago Torres for their paper, Factors Associated with Long-Time HIV Pre-Exposure Prophylaxis Engagement and Adherence Among Transgender Women in Brazil, Mexico, and Peru, Results from the IMPREP Study.
Our second awardee is Yunia Mayanja for her paper, Oral Pre-Exposure Prophylaxis Preference, Uptake, Adherence, and Continuation Among Adolescent Girls and Young Women in Kampala, Uganda, a prospective cohort study.
And our third award goes to Nicole McCann for her paper, Prevalence of Undiagnosed HIV Among Children in South Africa, Cote d'Ivoire, Zimbabwe, a model-based analysis to inform pediatric screening programs.
Our 4 JIAS Impact Award winners will now share their perspectives by video. We congratulate them and appreciate their contributions to the journal.
Diallo and I would like to thank JIAS for selecting our paper for the Impact Award. This is a wonderful recognition of our colleagues and of the years of work that went into the IMPREP study.
My name is Tiago Torres, and I'm a public health researcher at the National Institute of Infectious Disease at Fiocruz, Brazil.
My name is Katharine Bar. I'm an associate professor at USC and an associate researcher at UPCH in Peru.
IMPREP was the largest PrEP implementation project in Latin America and evaluated the feasibility of oral PrEP for over 9,500 MSM and transgender women participants within the public health systems of Brazil, Mexico, and Peru.
Our paper focused on the trans women enrolled in IMPREP. While we were able to enroll trans women interested in receiving PrEP, their long-term engagement and self-reported adherence were low. These results highlight the need for trans-specific interventions and the potential for long-acting PrEP.
I'm Dunia Mayanja, a research scientist for the MRC UBRI and NSH team in Uganda Research Unit. I am honored and humbled to receive the IAS Impact Award 2023. Our project was an oral PrEP implementation project among adolescent girls and young women. We found high willingness to take oral PrEP, however, actual uptake was low. Uptake was significantly higher among those who had most sexual partners and those who preferred oral PrEP to other biomedical prevention methods. Laboratory assays on plasma showed very low adherence, which did not match self-reported adherence data. 1/3 of the young women discontinued PrEP. We find that young women are willing to take oral PrEP. Oral PrEP works and is still the only available biomedical prevention option in many settings. Young be supported to take and adhere to oral PrEP as we wait for more prevention options. Thank you.
I'm Nicole McCann. I'm a PhD student at Boston University School of Public Health, and I'm honored to accept the JIAS Impact Award. I'd like to thank my co-authors, including senior author Andrea Ciarnello, for her mentorship. In our study, we used the CPAC pediatric simulation model to project prevalence of undiagnosed HIV in children aged 2, 5, and 10 in South Africa, Cote d'Ivoire, and Zimbabwe in 2018. We projected 18 to 56% of 2 to 10-year-old children with HIV were undiagnosed, with higher rates in 2-year-old compared to 5 and 10-year-old children. We projected over 50% of children with HIV would die before diagnosis, with 80% of these deaths before age 2. We suggest that targeted testing is needed for children of all ages and settings outside of traditional pathways, and that routine testing for children under age 2 or of all ages in high HIV prevalent settings could be impactful. Thank you.
Thank you for your participation. Please consider sending your best work to the Journal of the International AIDS Society. This concludes the morning session. Thank you.